Look at a crowd of people drawn from every corner of the world, and the differences seem obvious. Skin that ranges from the palest to the darkest, hair that falls straight, or waves or coils tight against the scalp, the shape of a face, the set of the eyes, the height of a brow. For most of human history, we've read those visible differences as a kind of map, quiet proof that beneath the surface, the groups we call races must be deeply, fundamentally separate. Different branches, different stock, different all the way down. It feels like plain common sense. And for a very long time, no one had actually measured whether it was true. Then, a team of geneticists set out to do exactly that, to open up the human genome across the entire planet and put one simple question to it. Where does human difference actually live? Not the difference we can see in a face, the difference written into the code itself, between the groups we sort people into, or somewhere else entirely. The answer they published runs almost exactly opposite to what that crowd of faces would lead you to expect. It is one of the most quietly radical findings in modern biology, and most people have never heard the number. This is the story of one of the largest surveys of human genetic diversity ever assembled, the result it produced, and what that result does and does not mean about the differences we think we see. Stay with me to the end, and you'll understand something that a lot of people, including plenty who speak very confidently about DNA and ancestry, get badly wrong. It isn't complicated, but it changes how you read every face in that crowd. If this is the kind of question you like sitting with, consider subscribing, because it's the kind we ask here often. To see why the finding landed the way it did, we first need to understand what a geneticist even means by the word difference, because it is not the thing you're picturing. Line up the full DNA of any two human beings on Earth, a farmer in Nigeria, a fisherman in Norway, a weaver in Peru, and the two sequences are around 99.9% identical. Nearly the entire instruction set that builds a person is shared, unchanged, across the whole species. Everything we argue about, every visible trait, every ancestry estimate, is competing for room inside the remaining sliver, roughly 0.1%. That thin margin is what scientists call human genetic variation. And the real question was never how much of it there is, it was how that variation is arranged. There are only two places it can sit. It can sit between the big groups, meaning most of the genetic distance in our species is the gap that separates, say, Europeans from East Asians from Africans. Or it can sit within those groups, meaning most of the distance is the ordinary variation between any two individuals who'd be sorted into the same box. Those are two completely different pictures of what a human being is, and you can measure which one is true. You take a pile of genetic markers, sample people from many populations, and do the statistical accounting. Of all the variation in the whole sample, how much is explained by which continent someone comes from, and how much is just people differing from their neighbors? The technique has a name, analysis of molecular variance, but the idea underneath it is that plain. Partition the difference, see where it lands. The first person to run that accounting seriously was the geneticist Richard Lewontin in 1972. He didn't have genomes, nobody did yet. What he had were blood groups and proteins, 17 genetic markers he could read across a spread of human populations. He sorted the world's people into the racial groupings of his day and asked how much of the total variation those groupings actually captured. The answer was about 85% within populations. Only around 15% of the variation lay between the big racial groups, and a chunk of even that was differences between nations and tribes inside each so-called race, not the racial line itself. The categories that felt so total, so obvious, were carving up a small corner of the picture. It was a striking result, but it rested on 17 markers. A skeptic could reasonably ask, would it hold if you could read thousands? 3 decades later, that became possible, and a team led by Noah Rosenberg and Marcus Feldman, drawing on the Human Genome Diversity Project assembled by Luigi Luca Cavalli Sforza, set out to answer it at a scale Lewontin could only have dreamed of. Their 2002 study in the journal Science reached into 52 populations across every inhabited continent and read 377 genetic markers in each person, a resolution orders of magnitude finer than anything before it. The expectation, even among many of the researchers, was reasonable. With far more populations and vastly more markers, the sharp lines between the great human groups would finally resolve into view. More detail should mean cleaner borders. It didn't. When they partitioned all that variation, between 93 and 95% of it sat within populations. Only 3 to 5% separated the major geographic groups from one another. The finer they looked, the smaller the racial fraction became, not larger. Lewontin's 15% hadn't survived the upgrade. It had shrunk. Sit with that number for a moment, because it's genuinely counterintuitive. It means that if you picked 2 people at random from the same population, 2 Koreans, 2 Nigerians, 2 Finns, the genetic distance between them would, on average, account for the overwhelming majority of the total genetic distance in the entire human species. The between group part, the part we've built entire histories and hierarchies on, is the leftover. The differences you can see, skin, hair, the shape of a face, are real, and they cluster by region, but they ride on a startlingly thin band of the genome, tuned by climate and sunlight over tens of thousands of years. They are the loudest 3%. They are not the story. Take the most charged, visible difference of all, skin color, and the genetics quietly dismantles its own myth. Pigmentation is governed by only a handful of genes, tuned over time by one main pressure: sunlight. Nearer the equator, stronger ultraviolet light favors darker skin, which shields the body's folate from being broken down. Farther from it, in weaker light, lighter skin helps the body make enough vitamin D. The anthropologist Nina Jablonski spent a career mapping how neatly human skin tone tracks the ultraviolet levels of the latitudes our ancestors settled. And here is the part that matters. The same trait arose by partly different genetic roots in different places. The lighter skin common in Europe and the lighter skin common in parts of East Asia don't trace back to a single shared light skin ancestor. Some of the key changes happen separately, in parallel, through different mutations. It's convergent evolution, the same solution invented more than once. Which makes skin color, the trait we've leaned on hardest to sort humanity into deep lineages, one of the worst possible guides to ancestry. 2 people can share a shade and sit on far apart branches. 2 people on the same branch can look nothing alike. To put that 3 to 5% in perspective, compare us to the rest of the living world. For a species spread across every continent, we are strikingly, almost eerily uniform. Many animals with far smaller ranges carry more genetic structure than we do. Our closest living relatives are a well-studied case. Separate chimpanzee populations in Africa differ from one another by more than all of humanity differs across the entire planet. We feel like a wildly varied species because we are exquisitely tuned to read each other's faces. A fraction of a percent of difference is enough to fill a lifetime of telling people apart. But by the genome's own accounting, we are one of the more genetically homogeneous large animals on Earth. The variety is real, it's just shallow and young and mostly shared. But here's where the same study gets used to argue the exact opposite, and where we have to be careful and honest. Because Rosenberg's team did something else. They fed all that data into a program that looks for clusters, without being told in advance who came from where, and asked it to sort everyone into a set number of groups. When they asked for 5 clusters, the groups it produced lined up, roughly, with 5 major geographic regions: Africa, Europe and the Middle East and South Asia, East Asia, Oceania, the Americas. To many people, sounded like a smoking gun. The computer found the races on its own. Doesn't that prove they're real, written into the code? The honest answer is more interesting than either side usually admits. The sharpest version of this challenge came from the statistician Anthony Edwards in a 2003 paper pointedly titled Human Genetic Diversity, Lewontin's Fallacy. His argument was mathematically correct, and it's worth taking seriously rather than waving away. Look at any single genetic marker, he said, and yes, most of its variation is within groups, exactly as Lewontin found. But look at many markers together, at how they correlate, and the picture shifts. The tiny between-group signal carried by each marker, stacked across hundreds or thousands of them, adds up to enough information to sort people into geographic clusters with real accuracy. Both things are true at the same time, and the whole game is holding them together. You can reliably estimate where a person's recent ancestors came from. That's genuine. It's the basis of ancestry tests and forensic panels. What you cannot do is convert that statistical sorting into deep, separate, ranked kinds. Classification accuracy is not biological essence. The gradient is still a gradient, even when you can make a good guess about which stretch of it someone came from. The clusters are real, but they are soft, and they are built from gradients, not walls. The program will hand you clusters for almost any number you request. Ask for 6, it finds 6. Ask for 3, it finds 3. None of those numbers is the true count of human kinds, because the program is drawing lines across something continuous. Most individuals came out as mixtures, partly one cluster, partly another, and the memberships shifted depending on which populations you sampled and how many. What the clustering captures is that people who live near each other tend to be genetically more similar, and that similarity fades smoothly with distance. It is detecting geography and ancestry, which are absolutely real. What it is not detecting is a set of sharp, separate, essential compartments. To see why, you have to look at how the variation actually spreads across a map. And when you do that, when you sample people not in a few far-apart clumps, but densely, step by step across a landmass, the walls dissolve. A study by David Sayre and Svante Pabo, and follow-up work from Rosenberg's own group, showed that if your sampling is even across geography, human genetic variation doesn't come in blocks at all. It changes gradually, continuously as you move, a little at a time, village to village, region to region. Geneticists call it isolation by distance. Neighbors have always traded genes with neighbors, so difference accumulates slowly with the miles between people, with no sudden jump where one race supposedly ends and the next begins. The apparent clusters in the earlier work were partly an artifact of sampling in scattered spots. Take readings only in Lagos and Beijing and Paris, and of course you'll see 3 groups. Fill in everyone in between and you see a gradient. The line was never in the genome. It was in the gaps between where we happen to look. Which raises a fair question. If it's all one smooth gradient, why is there any between-group difference at all? Where does even that 3 to 5% come from? The answer is a journey. Every person alive outside of Africa descends from small groups that migrated out of the continent tens of thousands of years ago, and each step of that migration carried only a sample of the diversity that existed back home. A subset left for the Middle East. A subset of that subset pushed into Europe and Asia. A subset of those crossed into the Americas last of all. It's a mechanism called the serial founder effect, like copying a copy of a copy with a little lost at each step. The consequence is that the deepest, widest range of human genetic variation still sits inside Africa, the source, and it narrows the farther a population's ancestors traveled from it. The 3 to 5% between groups isn't a ledger of separate essences. It's the faint trail of who walked where and how long ago. There's one more thread in that trail, and it surfaces in a lot of ancestry results as a small, surprising line. As the groups leaving Africa spread into Europe and Asia, they met other kinds of humans already living there, the Neanderthals. And in Asia, a more shadowy group we know mostly from a single finger bone in a Siberian cave, the Denisovans. They didn't just meet, they interbred. Most people with ancestry outside Africa carry a small fraction of Neanderthal DNA today, often somewhere around 2%. And some populations in Oceania carry a slice of Denisovan on top of that. It's a real, sourced piece of nearly everyone's genome. And it's a quiet reminder that even our deep history was never a clean branching tree. It was a network, with old crossings and merges written into it. The lineages we picture as separate were tangled together from the very start. Put faces to it, and the point gets sharper. The Human Genome Diversity Project holds real people from real communities. The Yoruba of West Africa, the San of the Kalahari, the Basque of the Pyrenees, the Karitiana of the Amazon, the Papuans of New Guinea, the Han of East Asia, the Sardinians of the Mediterranean, the Mbuti of the Congo forests. Among all of them, some of the deepest branches of human ancestry run through the populations of Africa, the San among them. Not because they are somehow older or purer as people, but because their lineages stayed closest to the wellspring of the whole species variation. And here is the fact that undoes the whole premise. Two individuals from a single one of these communities can be more genetically different from each other than either is from the average of a community on the far side of the planet. The box you'd sort them into simply does not capture the person standing inside it. It's worth pausing on what it took to gather those samples, because the project itself carried the tension of the whole subject. Luigi Luca Cavalli Sforza, the Italian geneticist who drove the Human Genome Diversity Project, wanted to capture human variety before migration and time could blur it, a snapshot of the species in all its breadth. It was a genuinely humane ambition. It was also fraught. Some of the communities approached were wary and asked hard, fair questions about consent, about who would own their DNA, about a long history of outsiders arriving to measure them and leaving with something. The project pressed on, more carefully for the criticism, and the panel it assembled became one of the most valuable resources in all of human genetics. The very data Rosenberg's team would later use to take the wall apart. The irony sits right at the surface, a catalog of human difference gathered at real cost that ended up showing how little of that difference divides us. This is probably where it stops being abstract for you. If you've ever spit into a tube and mailed it off for an ancestry test, everything we've just described is exactly what that test is doing. Those neat percentages, 40% this, 12% that, aren't reading a label stamped on your cells. The company is comparing your markers against reference panels of modern populations and estimating where along these same continuous gradients your ancestry sits. That's why the results can shift when a company updates its reference data, why two companies can hand you different numbers from the same spit, why there are always small, unexplained slivers that don't resolve. It isn't error exactly, it's the gradient showing through. You are being located on a smooth map that has no real borders, and then handed borders anyway, because borders are easier to read than a map. This is also where a lazy version of the idea goes wrong in the opposite direction. Saying race isn't a deep biological division is not the same as saying ancestry never touches the body. Sometimes it does. A handful of specific genetic variants really do cluster by ancestry, which is why certain inherited conditions, or the right dose of certain medications, can track with where a person's forebears lived. But those are precise, individual variants you can actually test for, not a global ranking of groups. The honest response isn't to go colorblind and pretend every difference dissolves. It's to trade a crude, ancient category for a precise, modern one. To ask about a person's real ancestry and their real genome, instead of reading a destiny off the surface of their skin. The old category isn't just offensive, it's low resolution. We can finally do better than it. So step back and hold the whole shape of it. The first careful measurement, in 1972, found most human variation sitting within groups, not between them. The largest fine-grained survey of its era, in 2002, didn't overturn that. It drove the point deeper, putting the figure at 93 to 95% within. Sample densely, and the borders between groups melt into gradients. Trace the gradients back, and they resolve into a single migration out of one continent. Every independent way of looking arrives in the same place. The visible differences are real. The ancestry is real and traceable. What isn't there, what the genome simply does not contain, is the wall. It helps to picture it as a spectrum. Think of a rainbow. The colors are completely real. No one pretends red and violet are the same. But the light itself is continuous, one wavelength sliding into the next with no break anywhere along the band. The names, red, orange, yellow, are ours. We drew them. Different cultures have drawn them in different places and counted a different number of colors because the lines were never in the light. That is what human variation looks like from the inside of the genome. A continuous spectrum of people, shading gradually across the whole surface of the Earth, onto which we have painted a handful of hard-edged names and then mistaken the names for the thing. And that is the quiet, radical center of it. Not that we are all the same, we plainly aren't, and the diversity is the point. But that the deepest genetic diversity in our species is the diversity within every population, carried by ordinary people who'd never be grouped together. We are far more a web of overlapping, shared inheritance than a set of separate branches. A spectrum, not a row of bins. The differences we can see are the thinnest, most recent brushstrokes on a canvas we hold almost entirely in common. 99.9% shared. And of the rest, most of it is the difference between you and your neighbor, not between your people and someone else's. None of this makes ancestry meaningless or history flat. Populations are real, migrations left real traces, and the gradients carry a genuine record of the human journey. What the largest surveys take off the table is the older idea underneath all of it, that humanity comes divided into a few deep, separate, ranked kinds. It doesn't. It comes as one continuous spectrum, drawn out across the world by a single long walk, with the boundaries added afterward, by us. So the next time you look at that crowd of faces from every corner of the Earth, you can let the differences be exactly what they are, real and beautiful, and almost entirely on the surface. The map is not a set of borders. It never was. It's a web, and every one of us is a knot in it. Thanks for watching, and if this changed the picture even slightly, subscribe and stay for the next one.